Hi everyone, today I'm going to be covering microbiology and today I'm going to be going over mutations. Okay, so what are mutations? Well, mutations are changes in genetic material. They can be brought about by a variety of subjects. It's a permanent change in the base sequence of DNA. And from that's also how you get natural selection, which is survival of the new, more favorable genotype. It's brought about by mutations. So there are different types of mutations. One such type is simple mutations, meaning they are silent mutations. The DNA base sequence causes no change in the activity of the product encoded by the gene. One nucleotide is substituted for another. It's usually at the third position. They could still code for the original amino acid, meaning that, which is why they're silent. Nothing could happen from them. They could be totally harmless. Or a new protein coded may function and nothing will change. It'll code a protein that's different. It's not what it was supposed to be. But really nothing happened because it does the same thing. Or it doesn't even function. Now there's also a base substitution mutations. And these are the most common type of mutation involving a single base pair. It's also known as a point mutation. So you'll see it written as both ways. Both. So a single base at one point in the DNA sequence is replaced with a different base, meaning that instead of, say, the mRNA, it could code for, say it was supposed to be a G, it turns into an A, and it creates a new protein. So, So it results in a substituted base pair, essentially, and it can lead to an altered protein in the grandchild cell. So kind of what happens here is seen right here in this little picture right here. So replication has occurred, and during DNA replication, a thymine is incorporated mistakenly instead of a guanine. So it wanted a G, but he got a T instead. So if it's not corrected in the next round of replication, adenine will pair with the new thymine, yielding an an AT pair, as opposed to a CG pair that was supposed to occur initially. So here's what it's supposed to look like, and here's what ended up happening, the granddaughter cell. Okay, so when mRNA is transcribed from the DNA containing the substitution, you get a completely different amino acid. So here, it really wanted a cysteine and a cysteine, but now it has a cysteine and a tyrosine. And this could cause changes in protein function, or it could cause nothing at all. It could still function properly, and we could move on from here, and everything would be okay. Another type of mutation is called a missense mutation. And this occurs when the base pair substitution results in an amino acid Okay, let me restart. Missense mutations, if the base pair substitution results in an amino acid substitution in the synthesized protein, this change in DNA is known as a missense mutation. So what ends up happening is the DNA, he didn't get coded right. That's what happened. And these can be very serious. This is actually how sickle cell anemia comes about. So here we have the original DNA template strand. Everything right here. So you have here's the protein there's the amino acid sequence. It gets translated, here's a transcription, here's the mRNA. Now, here's the missense mutation right here. The DNA template strand right here is where things change. What happens is, instead of the glycine, it becomes this one right here, a serine, amino acid sequence. So the amino acids changed, and it just changes what happens completely. And like I said, these can be serious. A nonsense mutation. Now, these are the really serious ones, because these create a stop codon in the middle of the mRNA molecule, and these can prevent creation of a complete functional protein. Now, whereas this one, the missense one, while these couldn't be serious, you at least still get the complete whole amino acid sequence, and a protein can be created. You at least get everything. Here, these create premature stop codons, and they prematurely stop coding the protein, and you could end up with something that was supposed to be there and now is not there at all. And from this, so that's one type of dangerous one. The other type of dangerous one is called the Frameshift mutation, and this occurs when changes in the DNA, as well as the base pair mutations. So it's kind of like a combination of the other two. So one or a few nucleotides are inserted or deleted into the DNA. So it can either get more A's than it needs, or it can lose some A's, lose some C's, lose some T's. And this causes a translational reading frame. And illnesses such as Huntington's disease occur from this because too many A's get added and it ends up improperly coding a protein. And now sometimes these actually code, and eventually you'll hit a stop codon. Or you could hit one too early. And then, of course, there's spontaneous mutations. And these occur when there's no mutation agents just available. They just happen. And it's because of a substitution or frameshift. And it's due to mistakes in DNA replication. Because remember, DNA replication, while very accurate, sometimes it just misses everything. And actually, the accuracy of DNA replication, it goes down with age. So the older the cell, the more likely it is to make mistakes. And so that is the quick overview of mutations, and I hope this helped clarify and made the different types of mutations that are possible, that could possibly occur, a little bit more easier to understand. All right, thank you.